So the work which I am going to discuss, it is done by one of my PhD students, Dhanir, who also got some training at Pittsburgh in the laboratory of Dr. Singh. So we all know about the incidence and the problem associated with cancer. Here, this is just a brief statistics. And in India also, it is growing every year. So this is the statistics for India. So what I'm going to take here, whether dietary habits, what we have today, can it have some influence or any influence on colon cancer or not? So simply, if we look at the development of the colon cancer, mostly it is independent of genetic factors. And what we have here, 85% to 90%, it is driven by the external or internal environment, what we have. And we also know about the risk factors, age, lifestyle, including intake of high-fat diets, alcohol, red meat, and so on. So these are already reported in the literature. We also know about the number of bacteria. You'll may be surprised to know that we have almost 10 times more bacteria in our body, what we have the cells in our body. And most of the bacteria, they reside in GI tract. Here's the distribution of the bacteria. So you can see 28% in GI tract, 26% in oral cavity. And these microbiota, they have also a job to do the fermentation of some of the components what we have in diet. So anaerobic fermentation of dietary fibers, it can produce several short-chain fatty acid, and butyrate is one of them. Short-chain fatty acid, if you look at butyrate, this is a very simple structure. And this is also a byproduct of several dietary fibers, starch, cellulose, and other things. And most interestingly, you can see that it can reach up to a very high concentration, up to 20 millimolar in our body. And there are several studies so far which are showing that it can inhibit the growth of cancer. So here we have tried to look at what has it for in colon cancer. And there is also a paradox called as butyrate paradox. Somehow it is very helpful for the normal cells. Butyrate inhibits colonic tumor cells, but it promotes the healthy colonic epithelial cells. And the mechanism of this is not well known so far. So we have used this compound and tried to understand how it works on the cell lines. Here we have selected two colon cancer cell lines, SW480 and HCT116. First, we looked at the effect on the cell growth. If you see here, time kinetics, 6 hours, 12 hours, 24 hours, in both the cell lines, there is a strong decrease in the cell number when we use butyrate 1, 2.5, and 5 millimolar. This is well below the 20 millimolar, which is known to be present in our body. We also looked at the protein, which is survivin here. You can see there is a decrease in the level of this protein, which is well known for the survival of cancer cells. Next, we looked at the cell cycle progression. If you look at, here you will find higher bars, which are indicating the cells being arrested in G2M phase. This is happening at 12 hours. When we looked at sub-G1 population, at 24 hours, they were very high. This is indicating that after cell cycle arrest, cells might be going for cell death, and apoptosis could be one of the mechanisms. Similar result was also obtained in the other cell line. Then we looked at some markers which regulate this G2M phase of regulation, well-known markers, cyclin B1, CDK1, and upstream is CDC25C. If you look at all these three proteins, they are decreasing when we are increasing the time and dose of sodium butyrate. So this suggests that the molecular alteration of these proteins may be associated with cell cycle arrest of these colon cancer cells at G2M phase. So here is the schematic diagram. CDK1, this is a serine threonine kinase, cyclin B1. It's a regulatory subunit. If it binds, it will become an active kinase, and cells will progress from G2 to M phase, from here to here. This is phosphatase, and it removes the inhibitory phosphorylation at threonine 14 and tyrosine 15, and that makes this active kinase complex. So we observed that this butyrate has efficacy to inhibit the cells in G2M phase, and that could be likely by a downregulation of these molecules. Then we also found that this is associated with apoptosis. As you see here, percent apoptotic cells, they are increasing with the sodium butyrate treatment in both the cell lines. Next, we looked at some apoptotic markers. They were also showing the favorable results, which indicates that this treatment is causing apoptosis. This was BCL2, it is getting downregulated, PARP cleavage, it's getting upregulated here, it's only cleaved PARP band, so all were in the favor of apoptosis induction. Next, we looked at the cleaved caspases, they were also increasing, so this also supports that cells are going under apoptosis. Next, we looked at what could be the role of caspase pathway. So we use inhibitor, which is very well known, pan-caspase inhibitor, Z-BAD-FMK. So when we use inhibitor here, part of the apoptosis was reduced. So this suggests that most of the apoptotic cells, they might be following caspase pathway for the apoptosis. In the meantime, what we were looking at, how to follow this work further, then my student, he came up with some ideas after reading this paper that mitochondrial fusion or fission, they may have role in deciding the fate of the cells. Then after this, we looked at the mitochondrial mass in these cells, and what we found that mitochondrial mass is decreasing when we are increasing the treatment with butyrate in both the cell lines. Next, we also looked at the mitochondrial ROS, and this was increasing. So this suggests that the mitochondria, which is active, it is decreasing, and the other one, which is not active or may have damaged membrane, that is increasing, and that could lead to increased RS production, which may be related to the cell death or apoptosis. Then at the same time, since we had already observed that this CDK cyclin B1, they are also playing role in G2M arrest, what we found here that there is a serine residue in DRP1. DRP1 is a dynamin-related protein, and it has some role in mitochondrial regulation. Actually, it causes fission of mitochondria. So what we found, this serine in case of human, it is at 616 position. However, it is conserved throughout all the species. So we focused our attention on this one. We also had one reference in this reputed journal that these may have some link, cyclin B1 and CDK, cyclin B1 and CDK1, is kinase complex. They may have some link with the cell survival. So this is hypothesis what we started working on. We had already observed that this complex is being inhibited, and now we have this information that DRP1, if this phosphorylation gets inhibited, its activity will be inhibited, and it may not go to form a clamp around the mitochondria to induce its fission. So in that way, there is a possibility that this compound butyrate may inhibit this process as well as fission of the mitochondria. Then we looked at the level of DRP1, and as expected, we found that there is a decreased expression of DRP1 after butyrate treatment. Its phosphorylation was also decreased. So this suggests that this compound has ability to inhibit the expression as well as phosphorylation of this DRP1. And at the same time, we also found that there is a protein called MFN2, which is for the fusion of mitochondria, and this was getting increased a little bit. So this suggests that fission might be going down and fusion might be getting increased. Then we also looked at the localization, where this protein is localized in mitochondria cytosol, because for its activity, it has to be translocated into the mitochondria. Then the fission of mitochondria can happen. And we found that the mitochondrial level is less when there is a presence of butyrate or treatment of butyrate. And this is overall decrease in the level of DRP1. Here we also observed that interaction of CDK1 and cyclin B1 is less, suggesting that butyrate is inhibiting the kinase activity. Then after this, we wanted to see what happens if we increase the expression of DRP1, since the hypothesis was that if you target DRP1, then mitochondria fusion may get inhibited. So if we overexpress DRP1, we should observe the result which could be otherwise. So in this case, when we overexpress DRP1, then we found that there is an increase in the number of colony or the survival of these cells. So this is, the black one is empty vector, and if you look at this highest bar, this is having overexpression of DRP1. Even in this condition, we found that the sodium butyrate, it has ability to inhibit the clonal expansion as well as survival of these colon cancer cell lines. So this suggests that if we have more and more level of DRP1, then we have more chances of having high proliferation and high survival in cancer cells, and that have been also clinically observed that some more advanced tumors, they have more expression of DRP1. Then in this case, we also looked at the number of live and dead cells. You can see there is increase in number of live cells with the DRP1 overexpression. And we also checked what is the effect it is having on the cell cycle distribution. So again, if you see, there is a decrease in the GTM phase population. This is the gray bar is for DRP1 overexpression cell line. So here, butyrate is able to induce GTM arrest, but when we overexpress here DRP1, the label got down. So this suggests that compound might be working through DRP1 also for inducing GTM arrest in these cancer cells. We also looked at the interaction of P53. So what we observed that DRP1 overexpression downregulates the expression of P53 as well as P21, and also there is an enhancement of mitogenic signaling which was measured in terms of phosphorylation of ORC1-2. So this tells that DRP1 overexpression can also leads to increase in the survival signaling of cancer cells. Then we tested this one with the inhibitors. Here P53 alpha is P53 inhibitor, ZVAD is caspase inhibitor. And what we found that decrease in P53 increases DRP1 level. So there is a vice versa control. If you target DRP1, then you can affect P53. If you target P53, you can also affect DRP1 level. And inhibition of caspases also increases DRP1 level. Interestingly, when we were starving the cells during the experiment, what we found that it is also giving the similar results. Whether the cells were treated with butyrate or there were starvation, results were similar, and we also observed that interaction between DRP1 and P53, which also happening during the starvation, it was observed with sodium butyrate treatment. So this may be the likely mechanism how DRP1 can be inhibited and cells can go under apoptosis through butyrate treatment. So overall, whatever experiments we did, this is the summary of the molecular observations. What we found that butyrate, it is causing GTM phase arrest here. Now we started looking at the mechanism, what could be the possibility. Then we found that this complex is being inhibited, CDK1, cyclin B1, and the phosphorylation and expression of DRP1 is also getting inhibited. And butyrate is able to increase P53 and also activate caspase pathway. So this is also linked with the DRP1. And if this process is happening overall, this could lead to the inhibition of fusion of mitochondria. Then there was a data for MFN. So this was observed to be increased after the butyrate treatment. So this can also lead to the fusion of the mitochondria. And overall, these markers were observed. So survival markers, they were downregulated. And those which were favoring apoptosis, they were upregulated. So that could be the possible mechanism for apoptosis induction. What we observed in overexpressing cells that they have more stemness property. So you can see the colonosphere formation here. DRP1 overexpressing cells, they are bigger in size. This is the data. and also there were more spheres when we expressed DRP1. So it is also increasing the stemness of colon cancer cells. And there was another experiment for different generations. So we observed here that DRP1 increases the regeneration capacity of spheroids, which is prevented by butyrate. Since we know now that butyrate acts post-translationally on DRP1 to inhibit its phosphorylation and expression. So this is the combined observation. There are synthetic inhibitors of DRP1 phosphorylation, MDIVA1. And now we found that butyrate can also inhibit DRP1 phosphorylation here at serine 616 position. And this is being regulated by cyclin B1 CDK1 kinase activity. And this is required for cells to progress from G2 to M phase. If we can inhibit this, we can inhibit mitochondrial fission. If the number of mitochondria is less, then most likely these cancer cells will not pass from G2 to M phase, and they will be arrested. Once they are arrested, then they may go for cell death. So here is the summary of the work. Butyrate inhibits mitochondrial fission, and it also reduces the energy efficient mitochondria as well as its mass. So in that way, it can lead to energy starvation, and this will result in cell cycle arrest and apoptosis in cancer cell. And thus, the dietary fibers, which we all hear all the time that they may have protective effect in our body, this could be one potential mechanism, what I discussed, through which dietary fibers can protect us from cancer development. Thank you so much.